Okay, here we are back again. I haven't gotten your exams graded yet. I have lots of other stuff I've had to deal with. I really try hard to get assignments and grade exams back, like the next class meeting. But that's just not going to happen this time. But I'll definitely, plan to have them back at the end of class next Tuesday, and we'll talk about them. And I'll have an entry key video, that you'll be expected to go through. And but we had our first exam, and you can see this is a hard course. And I think you could also probably appreciate that when we just stop and talk about any specific topic, concept, it's understandable, it's physical, it's accessible, it's a cause and effect relationship. The problem is just that there are lots of these pieces. We want to put as many pieces of this puzzle together as we can because they all resonate together. With one piece, you start to get an appreciation of, oh, this is just a very physical story like that. And so we're going to roll forward with that. And so here's where we're at. This week, I already posted and let you know before the exam that we're going to be talking about the next chapter, chapter 17. And we have a cell cycle assignment that I posted on Tuesday when you were taking your exam. That's going to be pictures that are going to be due by next Tuesday. So we got to roll forward in our course. And then I put this up here. This isn't due for a while. It's due a week from Tuesday. But it's another assignment. And I wanted to show you that because we haven't said that chapter 18 includes both the program cell death that we've talked about a little bit, and also has parts that talk about stem cells. And so that's why this stem cell assignment is up here. And you'll see the story of stem cells really matches and mirrors all the stuff we've been talking about in our course. And I'll kind of provide a foreshadowing example of that in class today about stem cells. So there's that. And then, in fact, exam 2 is a month from today. So Thursday, the 26th of March. And so we're going to roll forward. And I've already said, and we know what it's going to cover. It's going to cover chapters 17, 18, and 19. So let's just look at, a roadmap of what we're going to do the next semester. So our next month for exam 2. And I should mention that exam 2 it just includes the knowledge that we built up from here. So, there are examples going to be MIC. We're going to, continue to do that. But it's not like you need to go back and learn all, or review all this stuff. But it will be important to go over your first exam results. Yeah, Don Diego?
The syllabus is saying that we have exam 224.
Yeah, and so thank you for pointing that out because I updated that, and I talked about that, a couple weeks ago. And I just checked this morning too, and I agree, it had that. But then I updated it again. I dragged the updated syllabus back in there again, and it was showing me that. So you just looked, and it's still the 24th. Yeah, okay, I'll fix that again. It's weird. I don't know why it looks like it lands, and so I have to, figure that out better. Because, for example, it's showing the third exam. We slightly moved that exam, but it's showing that updated date, and for some reason, yeah. So thank you for checking that, because that's wrong. It's this date, Thursday the 26th, and I'll figure out how to get it to stick on the syllabus. Okay, on the syllabus on Brightspace. So thanks, Don Diego. Okay, and then there's this chapter 18. It's about program cell death and some stem cell biology like that. And those fit together, we'll see in a kind of a way, the stem cell biology is really a relatively new and exploding field of biology, so where to put it in this textbook, and it fits there well. It looks a little bit curious at this point, but it'll fit in well. And we've already talked about why would program cell death fit in here, and the answer is because a lot of cancers have two things going on. Especially they're growing out of control and they're failing to die. Because healthy cells have mechanisms to say, oh, if you're getting out of line, you should just go through this apoptosis and die. Okay? And then chapter 19 is about cancer biology. Well, we've been doing this all semester. So see oncogenes And we talked a little bit already about the idea of tumor suppressor genes. We'll have some really good examples that come up in this cell cycle chapter. And then molecular approaches to cancer treatment. So that tamoxifen with breast cancer and that cetuximab with the ERB cancer biology like that. It's really stuff that we've already learned, but it's just adding to it. In addition, it has this first section that is what you would think we would learn when we talk about cancer. So, you know, melanomas and carcinomas and some biology of cancers and cancer tumors and you know, cancer prevalence and stuff like that. But it really, we're just rolling with the kind of nuts and bolts details of it and how those causes of cancer can be used in a diagnosis. So remember that the FDA approved cetuximab, but only if you could also show that it was a wild type RAS in that tumor. And so there is a lot more of those molecular diagnostics, just categorizing cancers and different types of cancers based on their cause. And once you've done that, you move on to, well, can we find a treatment for that specific molecular problem? Like that. So that's where we're heading, and that'll be our second exam. And then for the rest of our course, we loop back into earlier chapters in the textbook. All right, great. So chapter 17 is really focused on eukaryotes. So of course, all life on Earth, the cells divide. So it's about cell division, but it's about eukaryotic cell division, and mostly about the mitotic cell cycle. So in the assignment video for next Tuesday, I explain the real simple differences between meiosis and mitosis. And we're talking about mitosis. That's what's happening everywhere except for in our ovaries and testes in our body. So all of our brain growth, right, when MYC is stimulating the proliferation in our bone marrow, that's the mitotic cell cycle. When our skin cells are healing under a scab stimulated by DGF, that's the mitotic cell cycle. So lots and lots of stuff like that, okay? And we can think of chapter 17 as divided into really these three parts. And we'll dive into them, but it's really three different stories like that that all fit together. So regulation of entry into mitosis, we start with that, then regulation of what's called start, and then the phases of mitosis. And our book covers all of those. So we're really talking about our book. We're just trying to talk about it in a complementary way. So it's really important to read through chapter 17 and bring in questions about confusion about it like that as we learn about it in class as well. Okay? So remember, this course is just really a pumped up Bio 105 intro bio course. And so I like to show this in intro bio, and it works really well here. So here's the cell cycle. And it has G1S, G2M, the famous phase of the cell cycle, like that, okay? And this shows 3 points in this cell cycle that we're going to talk about, those red things. And those red things are like gates or blocks or red lights, like that. And you can make the analogy of a washing machine, like that. So this is where you put your clothes in and press start on a washing machine. And then it goes through, you know, fills it up, wash it, rinses, spins it, and then spins it out, and it's done. It's back at start, you empty it, put more in, and start. And this is like a red light, and cells are waiting for things like PDGF and other growth factors to turn that light green. Yeah, okay? So that's our number one thing we're interested in, but it's really complicated with lots of checks and balances. It's also called the G1 to S transition. So there's G1 and there's S, and the cells are waiting, and they're waiting, and they're living. They're living, they're doing their thing, but they're at a red light and just waiting. Okay, like that. So for example, differentiated cells, our muscles, our brains, they've stopped dividing, they've stopped going through the cell cycle. They're sitting there at the G1 to S transition at start. They're just sitting there like that for the rest of your life, yeah? Those cells that are stimulated to grow in response to testosterone and estrogen during human puberty and vertebrate puberty maturation, those cells in a child are sitting, waiting at the red light, and they grow during adolescence. So the red light can be a temporary thing, it could be a permanent thing, like that. And so that's by far the most significant point. To understand it, we go over here to something that's called the G2 to M transition. So there's a red light right there, and a red light is just a DNA damage red light. And so cells should not divide, that's M phase, if there's DNA damage. So let's wait there and wait until the DNA gets damaged, gets corrected. So this is often called a checkpoint, because you're checking. And it's a DNA damage checkpoint, and that's where we're gonna start today. That's what we're gonna talk about today, is that, because it's a simpler little circuit of, detecting DNA damage and stopping the cell cycle. It's a little simple biochemical pathway that does that. And it has similarities to start, but it's just much simpler and satisfying. So a DNA damage checkpoint. Notice that that's called a checkpoint, and up here, this is called a control point, like that. And the control point is when the cell looks outside of itself, right? The washing machine is waiting for you to push the button. That's an external factor, like PDGF, that's gonna say go. These checkpoints are internal to the cell, where the cell is just saying, how are things going? Well, if there's DNA damage, I'm gonna stop. And then the third one that we're gonna talk about here is in the middle of M phase, and it's called the M phase checkpoint by some people. And I'm just gonna write its other name, because we're just gonna talk about it right now, and we're gonna be done with that for third one. It's a checkpoint, and it's called the spindle assembly checkpoint, or the M phase checkpoint. And the spindle that's being referred to is called the mitotic spindle, and that's where microtubules are bound to the chromosomes and get ready to pull them apart. So we're getting those chromosomes lined up in mitosis and in M phase, in metaphase, and lining them up and getting ready to pull them apart. So that's the spindle. The mitotic spindle is that assembly of chromosomes and microtubules and centrioles to set that up. And that's when the cell can stop in M phase. And it stops if that sort of that chromosome bookkeeping is not set up right. And it'll just, it's like a red light where it's just like, wait, this is not good. Okay? And the reason we're going to talk about it is in lots of cancers, this M phase checkpoint, the spindle assembly checkpoint, is often defective in cancers. And that's just another example of how cancer cells are just screwing up, just messing up. And cancers, as a result of this, cancers are often, so a cancer cell in a tumor, cancer cells are often said to be aneuploid. And that means wrong number of chromosomes. And it's a result of a defective spindle assembly checkpoint. So cancer cells are just messing up. They're not under the usual controls, either within the cell or within the body. And that's all we are going to say. Our book talks a little bit about the spindle assembly checkpoint, but that's all we'll say about it, unless it comes up and you want to ask questions about it and stuff like that. And so that's our third example, and it's a checkpoint. And these fit nicely into the washing machine analogy, because a washing machine is like a cell that's just hanging out. And where it's hanging out is at start. And the washing machine requires your finger, like a growth factor, an external signal to say go and to get it started. And then it's just going to go through, and if everything's okay, it'll just go through all the way back here and be back at start. and you take your clothes out, put more clothes in, press start. So that's what the cell cycle is, and that's why it's drawn as a circle, because it's a repeated series of the same steps. So you might know that if you leave the lid up on the washing machine, it goes part way and it stops, and it will just sit there forever until you put the lid down. And that's an internal checkpoint. And why it stopped is because it considers it's dangerous to have the lid up. Maybe your baby or your cat is gonna fall in before it does its fast spin, like that. So it's a safety step. So it's a really good analogy. Another one that you might have never experienced is if the washing machine load is out of balance, it will start moving around in the room and it'll stop and just wait for you to figure out what's wrong, like that, you know? And so that's like that spindle assembly checkpoint. You're just supposed to stop, and it's during the spin cycle, like that. So it's a realistic analogy of what's going on in this eukaryotic mitotic cell cycle. All plants, all animals, all single-celled eukaryotes go through this, okay? So again, we're already done talking about the mitotic spindle assembly checkpoint. That's what we're really interested in. This is what we're going to talk about today. And our book does that also, okay? So again, this PDGF is like a finger that's going to push start and make it go. And we know how it does it already, right? The PDGF binds the PDGF receptor, blah, 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 you know, RAS, RAF, MAC, ERK, ELK. ELK is going to be a transcription factor that activates some genes, and we're going to see that one of the genes can be MYC. We saw that already. And we'll see in the last chapter 19, there are other transcription factors that can also activate this, and we're going to make an essential component to that cell cycle mechanism. So it's a biochemical reality, right? We're using analogies, washing machine, red light, keep using them, but there's just a very straightforward biochemical explanation for what's going on. And what's going on is, to get through start, there's a rate limiting step. It's not the step we've been talking about, you know, not the step in transcription. It's a step in the cell cycle machinery that's missing and needs to be present or active to go. That's also true over here at the G2 to M transition. There's a rate limiting step that we have to correct if there's DNA damage. The rate limiting step has stopped. So that's what the red light means, is there's a rate limiting step, and to turn the light green, you have to overcome the rate limiting step. Like that, yeah, okay. And so here's that pathway, and that's what it's doing. It's gonna activate transcription, and products of that transcription are going to turn the start light green, and the cell will go into the cell cycle. So this is a fair drawing. I know it looks complicated, but you can see it. Under any scab we have, PDGF binds here in the skin cell, and it activates gene expression to get through that. That's what's going on, okay? Now, what we could talk about is also, we learned about the woman who had skin cancer. That pathway is disrupted. She's got skin cancer not under a scab. She just accidentally has that pathway activated all the time. So that tumor has activated RAS, RAF, MAC, ERK, ELK. There's going to be some MYC in those cells of that tumor going for it like that. Yeah. So both our normal health and also, you know, problems like that. And so I want to talk a little about this in that same assignment. We learned about the concept of the mitotic index. And the mitotic index is actually an indirect measure of how long the cells are sitting at start. So let's just talk about that for a little bit. So what we could do is, and I talk about it some in that assignment too, because what's going on is that you can think of, say, 100 skin cells. We'll use that as an example. And you can think of them as 100 cars. And when they're sitting at the red light, the red light doesn't turn green for all the cells. Each cell is responding to its own red light. And so if you look just at your normal skin right now, it has a low mitotic index. And what would it look like if you had 100 skin cells? almost all of your normal skin cells are parked here, waiting for their turn to go through a red light. It doesn't turn green for everybody, not like a regular street, it just turns for each cell. And a cell gets to go every once in a while, just randomly. And so there would be cells scattered throughout the cell cycle in our normal skin, and then they get around there and they stop again and wait for their red light to turn green. And you can see by this that some of the cells will be in mitosis, just by chance. And that's how we get the mitotic index. It's a percentage, and the more frequently the light turns green for individual cells, the higher the number, frequency, the ratio, the percentage would be that happen to be in mitosis, like that. Okay? So that's that concept. And each of the cells is waiting. So our regular skin cells have a low mitotic index. Under a scab, mitotic index increases. What does that mean? It just means that more frequently, individual skin cells are seeing a green light and going for it. They're going and they just go around the cell cycle. And so under a scab, we'd see more cells scattered along there. And that would be true for cancer too. The issue when the mitotic index goes up is a change in start. And the frequency that cells are, in the population of cells, are allowed to go through start and go through the cell cycle. Okay? So that's what's going on with that. The mitotic index is easily measured because you can see the condensed chromosomes. Mitosis, in the M phase, the chromosomes are condensed and visible under a microscope. So anytime you just look at skin cells under a microscope, count 100 and say how many of those can we see the chromosomes in. Those cells are in M phase, that's the mitotic index, and it's an indirect measure of what we really care about, and that's start, and how frequently a cell gets to go through start. PDGF is going to act on that point, and it's just going to turn the light green more frequently for these individual cells. Yep, okay. So one of the stories that we'll bring along with us is MYC. So MYC is a transcription factor, and MYC can activate gene expression of cell cycle factors that are the rate limiting step at start. So MYC can do the things needed to make the cell go, to make that red light green for that cell and go around the cell cycle, okay? So it's a good example, and this is a good example. It's almost an oversimplification because of the way we aren't talking about all the checks and balances. But it's a fact that MYC is activated under a scab, and it's helping to get the cell cycle turned green at a higher frequency. So it's a good analogy, but it is, we're not skipping over anything. We intentionally picked an example before the course started where the story is as simple as possible to go from extracellular signaling factor like this to making the cell go through the cell cycle, okay? So in that context, I just want to do this. I said I want to talk about one example in stem cell biology today. So this will be on our second exam, but it's not until chapter 18, okay? But it's cool to hear about because of where we're at with our learning that we've done in our course so far, okay? So it's a stem cell biology story. And people are interested in stem cells in medicine because maybe we could get some stem cells and we could help with an injury. Like maybe you injured your spine in an accident or your brain, and maybe we could make new cells for you in different parts of your body. People that have kidney disease, kidneys can't heal. They're waiting around for a transplant. Well, stem cells have the possibility of maybe making a whole new kidney. So it's a big deal, right? Like that, okay? So here are these two scientists, and they won the Nobel Prize 15 years ago, okay? And this guy, his work was done 50 years before that. And he was the first scientist that did an experiment to demonstrate genomic equivalence. So that's what this is going to be based on, right? Is genomic equivalence provides the possibility, theoretical possibility, that we could take a cell from you and make a new kidney for you, it wouldn't be rejected because it's your kidney, like that. So that's what this is rolling toward, and genomic equivalence provides that theoretical possibility. We already know what the problem is. The problem is, in those differentiated cells, they've methylated a lot of the genes, right? They use DNA methylation to turn off the other genes. We learned about that in the first videos in our course, right? So if you're gonna take a differentiated cell, like a muscle cell, and turn it into a kidney, you're gonna have to demethylate all those shut down genes. The other challenge is that these skin cells, brain cells, they've stopped the cell cycle. They're at that red light and they're done, right? Your brain cells are not gonna die again. So we'd also have to be able to take those cells and somehow get that cell cycle going again, okay? But the first scientist was a long time ago, just establishing that genomic equivalence, okay? And then this guy in Japan, just 6 years before he won a Nobel Prize, had an idea that is easily explained to all of you. It starts with genomic equivalence, and then it's like, how can I get those cells to turn into stem cells? So remember, in the first videos, we said, well, there is this MyoD, and if you misexpress MyoD, it'll turn any precursor cell into a muscle cell, like that. So, you know, all the genes are available, and you can change things around, right? So that gives a hint of how we could get things going, functional genomics, and then get genes going again. Then the next thing is to say is, remember this idea that the muscle differentiates, and it does that by expressing muscle genes. And I picked that example because you probably learned that muscle fibers have actin and myosin proteins. They're the contractile proteins, and there are lots of other proteins in that muscle fiber, like troponin and troponin T and so forth. So those proteins have genes that must be activated. And that's true for all of our tissues. They have all the genes, but they're only using what people call tissue-specific genes or muscle-specific genes, brain-specific genes, pancreas-specific genes, lung-specific genes. And so Yamanaka thought, given that, a stem cell must be expressing stem cell genes. For it to be a stem cell, it's being a stem cell. It's not being a muscle cell or a brain cell, it's being a stem cell. And so he decided to try to compare gene expression between a stem cell and a cell that's not a stem cell. And here's an example of that. So we talked some about this, and so we've already learned about the muscle satellite stem cells. And those stem cells, so the muscle satellite stem cells that we learned about at the start of our course, they're stem cells, and we'll learn more about them. They're in chapter 18, like that. But what we learned is that what they do is they divide when needed. They divide, and they're said to be self-renewing. So they'll divide and make another stem cell. And then they'll make a precursor cell that's going to become a myoblast and help make new muscles. And we know why that's needed, right? The muscle fiber can grow a little bit, but those cells have stopped permanently from the cell cycle, and the only way to get really much bigger muscles or heal muscles like that is to make new muscle cells. So see, it's this precursor and stem cell. And so this Yamanaka says, well, let's compare the difference in gene expression between those two cells. They both have genomic equivalence, they have all the genes, but they're starting to use them differently. This cell is starting to down-regulate the expression of stem cell genes. So in this cell, we'd expect to be able to detect what? We'd expect to be able to detect RNA, because the genes are being expressed, of stem cell genes. And what people call them is they call them stem cell identity genes. And that goes back to the muscle, the tissue story, right? Actin and myosin are muscle identity genes. That's what makes them muscle. You know, it's kind of almost like a circular definition, right? What's that? It's muscle. How do you know it's muscle? It's expressing muscle genes. like that. How do you know they're, how, whatever, however you'd be like, and then, what's expressed in a muscle genes, like that. That's what provides the identity, and it's literally be able to be detected in an experiment. Because remember, we said if a gene's being expressed, it's got to be transcribed, and that RNA must be present in the cell. It turns out the proteins would be present also, but it's easier to study all the RNAs at once than all the proteins at once. And so Yamanaka just made comparison, and he's looked for his lab in Japan, looked for genes that stopped being expressed as the stem cell stopped being a stem cell, like that. And then they said, let's swap the cis-regulatory region of those genes and make them be expressed. We could do that. You could do that experiment, right? How could we make those stem cell identity genes be expressed? Give them a new cis-regulatory region, right? So let's just say that, so everybody appreciates that concept. So through just rapid experiments, because the tools were there to look at all the RNAs, and they found these candidates, and they started out with 20 candidates, and they ended up getting it down to just 4 genes. And so, here's just an example of one of them. And this is a gene, and this is a gene that's a stem cell identity gene that's going to be expressed in a stem cell, and there it's being expressed, so it's going to enhance or transfer effects, stuff like that. And when it stops being a stem cell, that's going to stop being expressed. Well, we could force it to be expressed just by giving it a different cis-regulatory region. So here's a different gene that would stay on, and we just make a hybrid transgene like that, and just force the expression of these stem cell identity genes first in those precursor cells. Those precursor cells were a stem cell, yesterday, so it's not that hard. But then Yamanaka's group found that they could force the expression, or people say misexpress. And so I don't think we use that term in here. So people call this a misexpression study, and it just means we're expressing it, and we can define that. So this is misexpression. And just to say it, I'll just say it. We're going to use that term, but just to show you, there's another term that people use that's also a nice kind of term, but not kind of as good. People call it ectopic expression, and that comes from an ectopic pregnancy. Ectopic pregnancy is not happening in the uterus. It's gotten outside of the uterus. So you're getting pregnancy developing at the wrong place. So that's what they say, misexpress or ectopic expressing. We're expressing it in the wrong place. Now, we would say, because we know what cis-regulatory elements do, we would say, so misexpression is not a great name for this, because when we misexpress something, we're going to misexpress it in the wrong place, wrong time, or wrong amount, right? That's what the cis-regulatory region does, where, when, and how much. So the ectopic just seems to be place, but it's not. It could be wrong place, wrong time, wrong amount. So misexpression is that, and this is just an example of a misexpression study. What happens if we take these stem cell identity genes and we force their expression in a muscle cell? What happens? And the answer is they just turn into stem cells. And you could use them to make any cell. Yeah? And the name of those is, because they're made in a lab, right? They're not natural. And again, they're covered big time in our next chapter. They're called induced. That means, you're forcing them, kind of designing them like that. Induced, misexpressing, like that. They're called pluripotent. And that refers to the capacity stem cells. So it's letting you know these are not, really natural stem cells found in the body. They were created in a lab, just the way we described, just forcing expression of those, okay? Okay, so those 4 genes, we'll learn a little bit about them, not too much. And you don't have to scramble to write this down, but just to show you where we're at, we're talking about stem cells. So we know about what are called adult stem cells. This satellite muscle stem cells, that's an example of that. You probably have heard of embryonic stem cells. We'll learn about those in chapter 18. Those are natural stem cells. Induced pluripotent stem cells are real cells. You can use them, you know, they're alive. They're just not found in nature. And they got built, they're a transgenic cell. They got built by making an expression of genes and force the expression, okay? And so this again from chapter 18, and we don't have to focus too much on this, but this is them showing it, and they're naming the 4 genes here, and we're not gonna worry about the names of 3 of them, but the 4th one is MYC. So they can take any cell and make it go into the cell cycle by misexpressing MYC. So that's just like sick, right? Our brain cells are never going to divide again, but they have the genomic equivalents, and they're sitting at that red light at start for your whole life. And if you misexpress MYC, they just start dividing again. That's the role of MYC, is let's take these cells that are arrested and just make them go through that cell cycle. Because of course you have to do that with a stem cell, got to get it growing and dividing. The other 3, all 4 of them are transcription factors. That's not too surprising, right? You know, we only need 4 transcription factors to turn on a whole bunch of genes, right? That's what's going on here, you know. The other 3 are all involved in changing DNA methylation. And those are the 2 things you gotta do. You know, is, oh, you got genomic equivalents. Can you make that cell grow and form other things? Well, you're gonna have it be a stem cell. And you can turn it into a stem cell just by making it go through the cell cycle and modifying that DNA methylation to open up the possibility of, you know, a muscle cell can now be a chondrocyte, like that, you know, because it's got the genes to be a chondrocyte, a cartilage cell. And you just open it up, you can start doing it. So it's sick, it's just sick. And that's why that guy was awarded the Nobel Prize, just like that. They're just like, oh my gosh, this is crazy stuff, right? And lots of stuff that's exciting about that. But it fits here because I want to show you this. So this is from our book, and it's showing the cell cycle, and it's showing this start, and there are some different names for start, but start's a good name. So it's also called the G1 to S transition. It's also called the restriction point, and our author uses all those terms. Start is great, right? And this is showing this. This is really common. If you learned about the cell cycle in InterBio or any other course, there's a really good chance you learned, there's G1, S, G2, M, and G0. And G0 is drawn like that. And people draw it that way because you can have brain cells hanging out your whole life, not going through the cell cycle. Your kidneys can't heal because they're not going through the cell cycle. Not going to happen like that, right? And it's drawn like this. It's drawn like it's the cell's on a detour. And it's too bad that anybody ever talks about G0, because what's happening is those cells are just sitting at that red light for the rest of your life. They're not on a special alternative detour route. And if you could turn that light green, they go through the cell cycle. And MYC can do it. How does it do it? It activates genes that are rate limiting for the cell cycle in any cell, like that. Just crazy. Is this an oversimplification? No, you know, but yet weirdly, how can it not be? But yet the guy won the Nobel Prize for being able to just misexpress those 4 genes and turn any cell into a stem cell, which means make any cell go through the cell cycle. So, you know, just a little bit of what we're going toward with cell cycle stuff, okay? Great. So that's that. So, you know, G0 is a thing. You'll hear about it many, many times. It looks like it's a detour. It's not. It's just start. Start is just an amazing thing, and it's like a red light. It's like you got to push the button and make the light red. So here's the cell cycle again. We got to get back to that, talking about that, and driving toward, you know, the G2 to M phase transition, GNA checkpoint. So just from the start of the chapter, The original research that was done in this to make these discoveries was done in the yeast that causes bread to rise, and then the yeast that's used a lot in fermenting like beer and stuff like that. And the scientist that was working on the yeast that causes bread to rise was a guy in the United States, in Seattle, named Lee Hartwell, and his lab was at the Fred Hutchinson Cancer Research Center in Seattle. So he's working at a cancer research center, and he's studying the yeast that causes bread to rise. And what he found was orthologs, right, of all the cell cycle regulatory factors, and the way he found them is just by looking for mutants that failed to grow and sorting them out, you know, like that. And he won a Nobel Prize in medicine. He's studying yeast. So it's just a crazy story. The other scientists that studied beer were in England, and there was another scientist that won the same Nobel Prize with them that studied actually some clams, but it was like nothing like human stuff, but it was just all orthologs. And so that's the next thing we say is these switches are run by proteins, and there are orthologs of all those proteins in every eukaryotic cell. So if you do one of those blast searches for looking on a computer to look at relationships, you have no problem finding these in every eukaryotic cell. Honor. And you know, go ahead and do a blast search to look for insulin. Yeast doesn't have insulin or hemoglobin, it doesn't have myosin, it doesn't, you know, like that, but it has these cell cycle regulatory proteins like that. So that's what we're talking about, and that comes up here just, you know, one time to just give you that idea. And so we want to start talking on a focus point, this entry into mitosis. And that's when the chromosomes are going to condense, line up, and the cell's going to divide. It's called M phase, and it's here. It's just at the end of G2 and the start of M phase. And we're going to talk about that because, again, it's a pretty simple story, pretty accessible story, like that. Okay? And all right, so here are the action proteins that make the cell go into M phase. And the action protein is this one. It's called CDK1, and it's called cyclin-dependent kinase. So it's a kinase, right? So it's going to phosphorylate things, and it's going to cause the cell to go into M phase. So it's called cyclin-dependent kinase. Like that. And when that enzyme is active, the cell is in M phase. So it's a kinase, it's phosphorylating targets, and that causes the cell to be in M phase. It's always present in the cell, in every eukaryotic cell on our planet, all the time, and anytime it's active, the cell is in M phase. And we'll see how it gets activated, because that's the G2 to M transition. When DNA is damaged, CDK1 is inactive. When the damage gets fixed, CDK1 gets activated. And then this cyclin is just binds to it. It's a necessary protein to let the CDK be active. So cyclin-dependent kinase, great name. So it's just that protein, and if it's active, the cell's in M phase. Cells leave M phase, right? So the cell chromosomes line up, they separate, they start to decondense, they make a new nucleus around the two, you know, and then we have cytokinesis happening. When mitosis is ending, that cyclin gets degraded, and that means the kinase activity stops. So we'll learn about that. The way it starts is different than the way it stops. But again, that protein is there all the time, and when it's active, the cell's in M phase. If it got activated at the wrong time, the cell would just go into M phase. It's like the action protein, okay? So we're gonna learn its name. It's CDK1, and that's cyclin B. And in humans, and most other eukaryotes, I guess probably all other eukaryotes, each of those is in a gene family. So there's a set of paralogs. So there's a CDK gene family, and the members of the gene family are numbered. There's also a cyclin gene family, and those are given letters. And our book talks about the different numbers and letters, but we're just gonna learn these, CDK1 and cyclin B. And what we're gonna learn is this. Let's see, I have this on a slide coming up here. How far ahead? Too far ahead. Yeah, so this is a figure from your book, right? And so it's showing that at this well, maybe it's not showing it very clearly, but what it is showing is that there are several numbers, CDK1, 3, 5, 6, like that. And I'll just try to draw this out to give you a take-home message of this. And what it's like is this, that we're just talking about now 2 points in a cell cycle. G2 to M, DNA damage checkpoint is what we're focusing on first, and it's run by CDK1 kinase, cyclin-dependent kinase, and cyclin B. And it's just going to be, if you activate that thing, we're going into M phase. You know, so it's a good, simple switch for us to learn about. Over here at start, there are, I'm going to make them a different color, there are other CDKs. So they'd have different numbers, and we're not going to bother learning their numbers. Like that. So these are also CDKs, but not CDK1. So that's how we're going to learn about them. That there's a really good story here for cyclin-dependent kinases, and cyclin-dependent kinases control entry and presence in M. Over here, some other CDKs of that same gene family control entry into start, in this start of the cell cycle. But don't worry about those numbers. Similarly, there are cyclin-dependent kinases. So over here, there are some cyclins, and it's not cyclin B, you know, so there's some cyclins, and they have different letters, D and E and so forth. And we're not going to worry about it, but there's a key concept, and that is that these cyclin-dependent kinases play the central role at both of those. When we activate a CDK, that's when the action happens. And that's going to be what's really tightly regulated. But we're not going to get all caught up in the various CDK numbers and cyclin letters. So for example, when MYC gets activated, it has lots of target genes, but often its most important target gene is one of the cyclin genes. And that's a rate limiting step at start, is you just don't have enough cyclin to make these CDKs active. But again, start is more complicated. So we go over here to learn about the DNA damage checkpoint to see, kind of introduce the examples. Students get confused by this, but remember, we're mapping this out like that. And, we could map it out in this room. And when we're at the G2 to M transition, we're over there by the door. We're at start, we're over here by this window. And there's different stories at the two places. One of the reasons that students get confused is that we'll learn about this DNA damage checkpoint, and then we're going to go over here and see what regulates start. And as we do that, we're going to mention a little bit about al-Soli. When the light turns green here, there's a DNA damage checkpoint there, and it's totally different than that DNA damage checkpoint. So I'm trying to kind of help you provide this anticipation what's confusing. So we're at our first DNA damage checkpoint. That's the key thing here. The way to think about start is this. We'll get to start, we care about start. So start's like a red light, and at the red light intersection, there's also a school patrol person with a flag. They can walk out, and that's this DNA damage checkpoint. So the light can turn green at start, but the cell could have to wait if there's DNA damage. And that checkpoint is really, really interesting, what happens. Because if the DNA damage check doesn't get corrected here, the school patrol person shoots the driver. The cells go through apoptosis. Here, over here, they'll just wait. They're just gonna wait, like the lid up on the washing machine. They're just waiting. But here, it's a different scene. And it's an interesting one because the cell is saying, I don't want to go into S phase. I don't want to start replicating DNA if there's any DNA damage. Yeah? And who doesn't care about that? Cancer cells. Cancer cells just lose that DNA damage checkpoint, and they just start getting lots and lots of mutations and just more and more trouble. Yep?
Did you say that the rate limiting step at start is not enough cycling?
Sometimes.
Okay.
We'll see when we get to start, it's complicated. But it can be that. And that's often what MIC does, is it helps bind to the enhancer of a cyclin G and make more cyclin. Yep?
So what's the rate limiting step of that PDGF?
Yeah. So PDGF is going to, go through that whole pathway, and that is going to involve phosphorylating ALK. And ALK is going to activate several different target genes. Sometimes, in our example, this is true, it binds to MIC and activates MIC. And so it's like PDGF is going to cause a genomic response in the skin cell, new gene expression, and it's going to include MIC, and MIC's a transcription factor that will then bind to cyclin and make more cyclin. And it's just, the start is complicated, but that's true, what I just said. That we can find specific examples where the story seems, like, okay, got that. Like there's a series of steps that go one after the other, okay? So again, we're over here, and we're just like, oh, there's some DNA damage. And when the DNA is damaged, we should not go into M phase, okay? So that little talk there about both, I was just doing to kind of foreshadow how students get confused. Because it'll be really nice when we focus on the G2 to M, and you'll be like, got that. And then we'll say, we really care about S. So, next week we'll start talking about S, and it will start confusing people. Because we'll say, oh, there's that DNA damage checkpoint. And be like, But remember, they're like talking over by the door or up by this window. Totally different stories, like that. Okay? Different CDKs, different cyclins. All right, so we'll dive into this checkpoint, okay? And so here it is in a figure, and the figure, I think, doesn't show up until chapter 18 now, I don't know. But this is the one that we're going to be talking about right now, and it's very straightforward, and it's just that much. So down here, that's the protein we care about, CDK1. And up here is some DNA damage. And we are at the G2 to M transition. And we don't want to go into M phase. We don't want the cell to divide because we want to fix the damage before the cells divide. Okay? So here's what we want to learn. If there's DNA damage, this protein gets activated. And this protein is called CHEK1 kinase. So we're just going to talk about the little pathway. at this G2 to M. If there's DNA damage, there are several steps in detecting it, but for us, it's going to activate this CHEK1 kinase. And when CHEK1 is active, it's going to phosphorylate a protein called CDC25. Okay? And just a side note on that, this CDC25 is named by a geneticist, and it's that guy, Lee Hartwell, at the Fred Hutchinson Cancer Center in Seattle, and he was just numbering these genes. And lots of the genes he identified have been given different names, but he found CDK1, he called it CDC2, and so forth. But it's just an interesting historical fact that he was just calling them cell division cycle mutants and just numbering them in his genetic screen, finding all these different genes that made proteins that were essential for proper cell cycle regulation. So CDC25, and anytime we say CDC25, we like to say CDC25 phosphatase, because that's what it is. It's an enzyme that can remove phosphates. And now, just to show you that in more detail, I'm going to go over here and draw it out a little better and show you this, that on CDK1, that's our action enzyme for mitosis, CDK1 has some phosphates on it at different amino acids. Look like that. And two of these phosphates on CDK1 are inhibitory phosphates. That means they keep the protein off. And the function of this CDC25 phosphatase is, it's an enzyme, right? And it's going to catalyze the removal of the inhibitory phosphates. So, remember I said, oh, that CDK1, it's in our cells all the time, but it's not active. It needs the cyclin B, but it also needs some activating phosphates. And right now, what matters is it has to get rid of those two inhibitory phosphates. And the person who does that, the enzyme who does that, is CDC25 phosphatase. If there's DNA damage, this CHEK1 kinase gets activated. So there's CHEK1 kinase. And our book talks about this too. CHEK1 kinase activates and it adds a phosphate to CDC25 and turns off CDC25. So if there's DNA damage in the cell at G2 to M, CHEK1 gets activated, CHEK1 phosphorylates CDC25, CDC25 fails to remove the inhibitory phosphates, and we're just parked at a red light. The lid is up on the washing machine until you get the inhibitory phosphates removed. How does that happen? The DNA gets corrected, CHEK1 kinase gets inactivated, the phosphate gets removed from CDC25 phosphatase, and it goes to work, and it takes the inhibitory phosphates off of CHEK1. And that's, it's a pathway, it's like, a set of things, like that happen, and they're either going or not. And that's it, Again, the detection of the DNA damage is a little bit of a complicated story, so we're not going to dive into that. We'll just say it's DNA damage. Yeah? Couple of questions.
Just to clarify, if there's DNA damage, CDC25 fails to take off the inhibitory phosphate.
Right. Yep. Yeah. And as long as there's DNA damage, the lid is up on the washing machine. It will sit there forever. It's not going to die. The cell's just going to sit there and be like, man, I'd like to see you get that fixed. And it's sitting there because the CDK1 is ready to go, but it has inhibitory phosphates on it that need to be removed. And that's the total circuit for that checkpoint. And I call it a circuit because, you know, the lid on the washing machine, there's a little button, you know, the lid has to press when it's down to say, oh, the lid's down. It's a circuit, a little electric circuit. This is just a little biochemical circuit that says, if there's DNA damage, we don't want to go to M phase. How are we going to do that? Well, we won't remove the inhibitory phosphates. In this case, there's plenty of cyclin B hanging out, so that's not the situation. So cyclin B is there, and it's, you know, cyclin-dependent kinase 1, right? So it needs that, it's there. You know, everything's looking good, but the inhibitory phosphates are there. We can answer questions about those activating phosphates. It's a totally different story, you know, but it's another, you know, story about its action. But it doesn't have any effect, any role in our story for the DNA damage checkpoint. Yep.
So can cyclin B only bind to CDK1 if inhibitory phosphates are removed, or
No,
does that
it
not
can
matter?
be, yeah, so say cyclin B can be bound like that, ready to go. I'm like, go, 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 go. But it just can't go without the inhibitory phosphates. Like that, okay? We're doing great. All right, and so that's the checkpoint circuit. Again, like the idea, you know, lots of us are not that interested in, you know, electronic stuff and how a washing machine works, but in fact, you know, when the lid gets pushed down on the washing machine, there's a little button, the lid presses down, and it completes an electric circuit, and the washing machine says, I guess we're okay to go into the spin cycle. This is just like that. It's a biochemical circuit like that, and it's like a little button, you know, and if there's DNA damage, that gets detected, CDK1 gets activated, adds that inhibitory phosphate to CDC25. CDC25 is just sitting there going forever, like that, until that gets removed. And what it's waiting to do is just remove those inhibitory phosphates so then CHEK1 goes. Again, CHEK1 is present all the time. Cyclin B is always present at the end of G2. Cyclin B just accumulates during G2. And so it's not, you know, it's never the problem in this G2 to M transition. When it's, you know, when the cell gets to the end of G2, there's cyclin B. The big deal is just those inhibitory phosphates, and that's it, okay? And, you know, these are kinases and phosphatases. You know, we've met them before, you know, and we know sometimes the phosphorylation will activate, sometimes inhibit. You know, we're seeing that going on here like that. Yep. Yeah, Don Diego?
Cyclin B can bind to CDK. We do have on CDC25, right?
It's cyclin B just binds to CDK1, and it's going to help activate it, cyclin-dependent kinase. So there's no interaction between CDC25 phosphatase and cyclin B. CDC25 phosphatase is heading toward those inhibitory phosphates on CDK1. Yeah. So, you know, okay, great, like that. And the next thing we'd want to say is, what happens when CDK1 gets activated? So what does that mean? You know, okay, it's going to go to M phase, but what is its action like? Okay, so let me just show you. There'll probably be some slides from the book, because the book talks about this. And again, this is the summary figure in the book, but we'll look at it again, because it's the, you know, the artists try to make a good summary figure, and they made it too complicated, because you can see we just want to focus on that DNA damage. This is a different kind of DNA damage at a different time, not our story. This is our story here. CDK1, like that, super cool. And it just goes like that. You know, and I mean, I'm shrugging, I don't want to like misrepresent it and be like, everybody understands this right away, you know, because you got to sleep on it. But you will, you know, you can understand it, because it's like, just, and I should mention, these proteins are just floating around inside the cell. Some of them are localized in the nucleus, and some are just in the cytoplasm, but they're just hanging out like that. Yeah. In this case, it's worth mentioning that when CDC25 gets phosphorylated, it is a cell story. And I just want to share that with you, because our book talks about it, and it's actually really surprising thing to see. So again, these proteins in the cell cycle, generally they're just floating around, right? But it turns out that CDK1 is almost entirely in the nucleus. That's where that kinase is that we're talking about. And when CDC25 gets phosphorylated and inactivated, it looks like that, and that's that CDC25 phosphatase. And the reason it's inactive is because it's being held onto, when it's phosphorylated, it gets held onto by a protein called 1433. And it keeps it excluded from the nucleus. And that's how you can't remove the inhibitory phosphates. Yeah?
Is that the same one that keeps FOXO out?
Dang it. One time I taught this course, and at this point I said, if anybody knows what we've seen 1433 doing before, they get a grade of an A for their final grade. And this student named Dan said, oh yeah, bound to FOXO. I was like, darn it. But yeah, Liana, yes, it's the same crazy protein. And if you look at the Wikipedia page for, what a dumb name for a protein, 1433. I mean, who named that? Some grad student, you know, down in the lab in the basement going, oh yeah. If you read the freaking Wikipedia page of what 1433 does, that's all it does. But it does. It has, you know, impacts on cell biology that's just crazy. Like, you're like, oh my goodness. Yeah, Chase?
I was going to ask the same.
Yeah, it's that same protein. And 1433 is a member of a little family of proteins. And they just have crazy cool biology. But that's actually what's happening here. You know, and it's very much like the FOXO story, where FOXO has to get in the nucleus to do its job. And this, well, got to get in the nucleus to do its job, like that, right? So that's that, yeah. So, you know, just kind of a detail, but kind of like, yeah, cell biology is cool. All right, so there's that. And so let's just look through a couple slides, see what else comes up here, because we might be, so somewhere I might have to slide to these slides because we're going to soon talk about START. And we have a few more minutes, but we're still in the G2 to M transition. So let me find these slides. Oh, you know where they are is, I think they're in the drawing assignment for, you know, next Tuesday is where I move those into. But let me just say that this CDK1 kinase activates two other helper kinases. So, and our book has this in a figure. So now we're going to say like, CDK1 gets activated. You know, the DNA damage is corrected, it's activated, and we're going to be in M phase. When CDK1 is activated, we're going to be in M phase. And the first thing it does is CDK1 has so many things to phosphorylate. CDK1. CDK1 has so many things to phosphorylate that it phosphorylates these two helper kinases called Polo and Aurora. And that's just a, you know, kind of random detail, but it's just that it has so many targets it needs to phosphorylate that it needs help. So it activates Aurora and Polo, and all three are really famous kinases because here's some things they do. So the chromosomes condense and become visible, and that's like a hallmark visual definition of M phase. In every eukaryotic cell, when it enters M phase, the chromosomes condense. That means they shorten. And the reason they shortened is because there are changes in the organization of the nucleosomes. So we learned about those nucleosomes. And the nucleosomes are helping organize the DNA. The DNA is wrapped around the nucleosomes. So people say DNA is like beads on a string, on a beads, or the nucleosomes are like beads on a string like that, so that's the DNA. And they're helping to pack the DNA into the cell. And those nucleosomes are made up of proteins that are called histones. And one action of these three kinases is to phosphorylate histones. And when histones get phosphorylated, they fold up in a different way, and they fold up and the chromosomes condense more. So remember, we saw euchromatin, that's open, that's acetylated histone. We saw heterochromatin, that's deacetylated. When histones get phosphorylated, they pack up even tighter. And they pack up to the degree that chromosomes get so short, you can see them. And it's just that. Like, is that an oversimplification? No. That's just check one and what it does. You know, like that. And so if you get check one, sorry, I said it wrong again, CDK1. So that's just what CDK1 does. That kinase is the action protein. And it gets help, but it just phosphorylates the stuff that happens during M phase. The nucleus disappears. That's because CDK1, Aurora, and Polo phosphorylate target proteins that are holding the nuclear membrane together. And the nuclear membrane just breaks up into little droplets. And so you see the chromosomes and the nuclear membrane generally disappear, chromosomes condense, and then at the end of M phase, we get the nucleus reforming, we get the chromosomes decondensing because CDK1, I don't know how I got hung up on that, CDK1 gets inactivated like that. So the video goes into that some. We'll follow up that on Tuesday. And then we're done with that, and then we'll move over, right? We've been up here by the window talking, and we'll move over by the door and start talking about the start. You know, and there's some parallels. Check, the CDKs are the big players, but it's different CDKs, different stories on start, okay? So we're gonna stop. Again, I apologize that I didn't get the exams graded yet for today. They're heading back, but I'll have them next Tuesday, and we'll roll on from there. And so remember, you've got that assignment. It's already on Brightspace. For the cell cycle to dive into more parts of that. Some different parts than we talked about.